right, everyone. Hello, my name is Holly de Rosa. I'm a graduate student collaborating with Dr. Amanda Kentner at the Massachusetts College of Pharmacy and Health Sciences in Boston, Massachusetts. Today, I'm going to be presenting data from our latest project, which sought to investigate the effects of maternal immune activation experienced during the mid-lactational period on maternal milk quality, parental care, and offspring development in Sprague Dolly rats. The neonatal environment requires a high level of maternal demand in terms of both breastfeeding and other forms of maternal care. Previous studies, especially the work of Dr. Michael Meany, have underscored the importance of these maternal factors on offspring development and behavior. However, their contribution as dynamic factors in animal models of early life stress are often overlooked. And so, as I'm sure we're all familiar, one way you can model early life stress is by prenatally exposing pups to maternal immune activation, or MIA. In our lab, we used the MIA model to understand the biological mechanisms associated with exposure to prenatal infection. We also explore how these biological changes in the brain contribute to the emergence of certain behavioral deficits and even some hidden talents in MIA-exposed offspring. And so again, I'm sure we're all familiar, but just to drive it home, this animal model entails injecting either the bacterial endotoxin lipopolysaccharide, LPS, or Toll-3 receptor agonist, poly-IC, on gestational 15 in most of our studies, and models many of the behavioral and physiological phenotypes associated with neurodevelopmental disorders such as autism and schizophrenia. Much less, however, is known about what MIA exposure does to pups after parturition. And so in one study conducted by Dr. Maria Bernardi's group at UNIP in Brazil, they found MIA exposure during the third day of lactation to be associated with a significant reduction in the number of ultrasonic vocalizations of male rat pups, suggesting that MIA may be affecting pups by modulating certain aspects of maternal input signals. And so in the present study, after breeding our Sprague Dolly rats, we left them to give birth, which was designated as postnatal day 0 or P0. On P1, we standardized our litters to 10 pups per litter. We then waited until P10, and on the morning of P10, dams were removed from their litter and placed into a fresh, clean cage located in a separate procedure room. And this was done to facilitate milk accumulation so we could collect the milk samples. 2 hours later, we briefly collected maternal milk samples. Sorry about that. At the start of the separation period, we first gave an IP injection of either 100 micrograms per kilogram of LPS or pyrogen-free saline. We then, 2 hours later after that, we briefly collected maternal milk samples and promptly returned mothers to their litters after collection. 2 hours after being reunited, we assessed offspring huddling behavior. We measured maternal behavior on the afternoon and evening of P10. The next day on P11, maternal care was assessed again in the morning. Milk was collected at the 24-hour post-injection mark, followed by another assessment of offspring huddling 2 hours after being reunited. After weaning on P22, we took one male and one female offspring from each litter and had them complete behavioral measures between the ages of P43 and P45. We assessed maternal milk quality through the use of several assays. First, we measured percent hematocrit, which is obtained by spinning the milk down and measuring the percent ratio of this fat to whey layer. Hematocrit is linearly related to the fat and energy content in milk. And we found that MIA was associated with a reduced percent hematocrit on postnatal day 11. MIA was also associated with reduced triglycerides in maternal milk at P11. Milk corticosterone concentrations were increased in MIA dams on P10, but this recovered by P11. Importantly, we looked at two different markers of inflammation in our milk samples in order to explore the possibility that the LPS or associated inflammatory cytokines were passing from the mother into the milk and then being consumed by the offspring. We found no significant differences in interleukin-6, however, between our LPS and saline dams on P10 or on P11. I just want to point out, though, that the presence of IL-6 in our milk samples was not surprising, given that IL-6 is responsible for typical mammary gland functioning, including involution and milk storage. Therefore, we also looked at lipopolysaccharide binding protein, or LPB, in the milk and blood plasma of our dams on P10. Since LBP concentration is directly related to the expression of LPS, The low LPB expression in milk suggests very low or negligible levels of LPS in maternal milk samples of both groups. In contrast, the high levels of LPB in our maternal plasma of LPS-treated dams suggest significant levels of inflammation and the presence of LPS in the blood. And so these findings both validate our MIA model and suggest that inflammatory signals are unlikely passing through the maternal milk and getting into the pups. Microbiome sequencing of milk samples was also performed. Linear discriminant analysis of effect size, or LEFC analysis as it's commonly referred to, was used to identify bacterial taxa that were significantly different between our groups. Blue bars indicate taxa that were significantly more expressed in the milk of saline-treated dams, while red bars indicate taxa that were significantly more expressed in the milk of LPS dams. Very briefly, a greater expression of Christiansonella, nailed it, has been associated with lower serum triglyceride levels, whereas lacnospiraceae and ruminococcosae have previously been associated with inefficient lipid and energy metabolism in the gut. Greater expression of ruminococcosae in sheep's milk has negatively been associated with milk fat content. And so overall, we think that these microbiome data might be demonstrating that MIA specifically is targeting milk fat and milk energy content, possibly through driving the differential expression of certain microbes. And so before I discuss offspring developmental outcomes that were associated with these altered microbial and nutritional profiles, I'd briefly like to talk a little bit about our findings regarding maternal care. MIA dams exhibited a significantly lower frequency of self-grooming on the night of P10, and this was overcompensated on the morning of P11. MIA dams also had a higher number of eating bouts compared to saline dams, which was most evident on the morning of P11. And so this would have been during the recovery phase following the LPS challenge. And so what I am trying to get at here is maybe some compensation. So we found no differences in licking and grooming pup-directed behavior on the afternoon and evening of P10. Oh, excuse me. However, on the morning of P11, again, these maternal care behaviors were significantly increased in MIA dams. So this, combined with higher levels of food intake, might suggest that MIA dams may be compensating for their reduced triglyceride concentrations in the maternal milk. We did not observe differences between MIA and saline dams in the amount of time spent on the nest, or in the frequency spent in this more efficient archback nursing posture. And so overall, we didn't see these drastic changes in maternal care following mid-lactational MIA. In order to assess offspring developmental outcomes associated with neonatal exposure to MIA, we assessed several behavioral measures across the early life of offspring. First, on P10 and on P11, we observed pup huddling, which has previously been described as a form of social thermoregulation. Pups from MIA dams huddled significantly less compared to pups from saline dams on P10 and on P11. Pups from MIA dams also formed fewer clumps compared to saline dams on P10 and on P11. And so since undernourishment has been associated with a reduced neonatal huddling in rat pups, and huddling efficiency may be related to serum triglycerides in newborn rats. Again, we're thinking that this reduced huddling might be reflective of the lower milk fat content we saw in MIA dams. In adolescents, male offspring of MIA exposed dams were significantly heavier than offspring from saline dams at P43. We think the increased body weight might be reflective of lower triglyceride and hematocrit levels we observed in the milk of MIA dams, since metabolic dysfunction is often seen in children and animals who exhibit poor nutrition early in life. With respect to adolescent behavior, male MIA offspring demonstrated deficits in pre-pulse inhibition. And similarly, MIA was associated with an increased mechanical allodynia threshold, but this was only in female offspring. Just to briefly talk about some next steps with this project. So I'm currently in the process of learning how to do Western blotting. It's been a real pleasure. I want to measure the expression of parvalbumin interneurons in the hippocampus of our offspring. They're commonly implicated in models of developmental perturbations, including the prenatal MIA models. And so with this work, I hope to reconcile some of our offspring behavioral findings. So those results will be soon to come. And so altogether, these results demonstrate that the effects of MIA are not restricted to the prenatal window. The impact on offspring behavior is likely driven by a change in the nutritional and microbial profile of maternal milk, and not by a direct exposure to LPS, other inflammatory molecules, or by severe deficits in maternal care. Lastly, we hope that this work underscores the idea that animal models of early life stress can impact both parents and their offspring, and the quality of maternal milk should be considered among the variables to be investigated in the future studies. And if you would like to see the full details of this study, as well as a complementary milk study that we put together regarding the effects of environmental enrichment on maternal care, milk, and offspring outcomes, you can use your cell phone camera to scan these QR codes, and they'll take you directly to our preprints. Before I accept any questions, I just want to thank MCPHS for their departmental support, as well as the National Institute of Mental Health for funding this project. Thank you so much, Dr. Amanda Kentner. You're an amazing, supportive scientific mentor. You're awesome, so thank you so much. And thank you for also assisting with the milk and tissue collection. I'd also like to recognize undergrad Hugh Tran for being an awesome undergrad for his assistance with behavioral scoring. And so with that, thank you all so much for your attention and for listening to my talk. I will take any burning questions.
Thanks, Holly. That was really great. Does anyone have questions? Oh, perfect. We've got a couple. Lonnie, you're up at the top of my screen.
Thanks. That was very, very interesting, and like a new perspective on the maternal immune activation work than we've heard from so far. I'm curious, so one of the dominant themes that emerges from a lot of the MIA work is how much variance we see in the maternal response during the prenatal experience. And so from your plots, I didn't see the underlying data, but I was wondering about how much variance you see in that maternal response when you induce MIA postnatally.
Sure, thank you for that question. Yes, as you said, I think the MIA model can be pretty variable. I can't say that this was any exception. Yeah, so I think dosing is obviously a big role into it, and our lab prefers to use the lower dose because it's not modeling that like septic shock, super mega infection that leads to hospitalization. We're more interested in more benign physiological responses. And so, yeah, I don't think this was an exception to that variability, but that's behavior for you. So.
Lara, you've got a question?
Yeah, I mean, wonderful talk. And again, echo what Lonnie said. I think it's such a interesting space to be investigating. I would never even imagine to start looking at that. And I think it's fascinating, all these findings. And it gives me a lot of pause to think about all the other things we're seeing in the work that we're doing. One question I have for you is just the timing of your assays. So you did everything at PMD 10 and 11, or a lot of your measures at PMD 10 and 11. Can you maybe mention this in your talk? And I apologize if I missed it, but can you provide some rationale as to why you picked those 2 days and why something so densely sampled?
Sure. So there's a few reasons for that. For one thing, litter standardization. So moms will give birth to obviously a variable number of pups. And so by P10, we hoped that everything was just kind of like ironed out and like more regular. But the larger reason was because we wanted to look at this mature milk phase. So you have the early milk phase is like P1, P2, and then this mature milk phase is more indicative of like P10 and P11. And so this is when you're going to see like your how do I say this? Sorry. In terms of like nutrition, right? And so like triglycerides, we also looked at a few other maternal milk markers. So we looked at immunoglobulins. Those were not significantly different, in case you were wondering. But we looked at a couple of different things specifically because it tends to peak at around this P10, P11 mark. And the LPS exposure obviously is supposed to kind of wean off in a 24-hour period. And so that's why we wanted to do a 3 and a 24-hour time point to see, like, at the height of inflammation, what does milk look like, and what does milk look like at that kind of like recovery period.
Very cool. And one last question. I suspect you normalized everything for time of day.
Yes.
But what are diurnal fluctuations and in any of these markers?
That's a great question. Yes, everything in this study was very like time dependent, and everything was done typically in the morning. But yes, you can get diurnal fluctuations. I know off the bat because I just read about this recently, but in triglycerides in particular, I'm not obviously Cort, I imagine, would be another one that would probably fluctuate with diurnal changes and whatnot. But yeah, we did our best to keep everything in the morning, so everybody was collected at around a similar time.
Nice. Fascinating stuff. Thank you so much.
Cool. Thank you so much.